First of all, it's a pleasure to be here at Laboratory Medicine Grand Rounds. As I was mentioning to one of the members of the audience earlier, just about everything we do in our work in cardiovascular epidemiology involves the laboratory. And all of it is interdisciplinary, and it's very collaborative. And the laboratory is an extraordinarily important part of our work. And so it's a pleasure for me to be here to share some thoughts with you this afternoon. Additionally, this is the 20th year of the Cardiovascular Health Research Unit. Bruce Basady, who's also a professor of medicine and epidemiology, and I put our resources together in 1991. And we've been working collaboratively with each other and with the larger group in our unit and with people across the country and across the globe since that time. And I just want to emphasize that everything that I'm going to tell you today is work that has evolved in a collaborative setting, and it's not my work. It's the work of groups of individuals, and in some cases the groups are extraordinarily large. And you'll see why, hopefully, as I go through some of the material. So let me just start by saying that the bottom line here is that population-based genome-wide association studies have identified novel genetic loci associated with a wide range of cardiometabolic traits. I'm not going to have time today to review the world's literature that covers all of the cardiometabolic traits that have been examined using this approach, as you'll see, but I'll give you a little tutorial to begin with and then some selected examples. While the newly identified common loci typically have modest effect sizes, that is, they explain a relatively small proportion of the variance in a phenotype or trait, they do provide clues to novel proteins and pathways involved in metabolism, physiology, and pathology. The findings of GWASs should be viewed as an initial step and only as an initial step in efforts to discover new biology. And in fact, the discoveries of loci associated with various traits raise novel biological hypotheses. Interdisciplinary research collaborations are needed to explore further the mechanistic hypotheses raised by these studies and to translate them into clinical practice. So in a nutshell, with those 3 or 4 sentences, I've tried to summarize what's been a huge global effort that in many ways has transformed genome science and genetic epidemiology and statistical genetics over a period of literally 2 to 4 years. So the challenge is to identify novel genetic associations, and I'm going to focus today on some cardiometabolic traits. We would want to be able to use this to help explore mechanisms that might underlie these genetic associations, and obviously the gold ring, or the ultimate goal, is to provide evidence for causal associations that can then be used to either estimate genetic risk or to identify targets for intervention. Classically, genetics has focused on families, and basically you start with an observation that there's familial aggregation of a particular trait. You then try to determine, is the trait heritable? If the trait is heritable within the family, then it could be due to shared genes or shared environment or both. In genetics, linkage analyses, first segregation analyses followed by linkage analysis, was the way this was explored to determine whether familial aggregation and heritability might be due to a mutation. And then there was fine mapping of regions of loci identified from linkage analyses in an effort to identify the causal variant, the mutation that accounted for that particular trait within that family. Now in genomics, what's happened is that with the sequencing of the human genome, which dates back now almost a decade, it was clear that there's tremendous sequence variation across the genome, and there was an opportunity to take advantage of this sequence variation to identify single nucleotide polymorphisms, that is, variants in base pairs that occur with a frequency of 1% or greater, that might tag different regions of the genome, particularly as it relates to common sequence variation. And then the genomics has now moved forward beyond tag SNPs to sequencing in large numbers of individuals, either using targeted sequencing, whole exome sequencing, or eventually whole genome sequencing will be used broadly or more broadly to detect rare variants. So most of you I'm not sure how familiar you are with some of these terms, and so I thought I would go through them and maybe put this work into context. So this is really a marriage of population-based studies and genome science, and it involves epidemiology, statistics, and it also involves metabolism, and it involves people who know phenotypes. So basically in population-based studies, you can think of observational studies as either using a case control design or a cohort design. In a case control study, you obviously start with the outcome, and you might look at a single outcome like myocardial infarction, which is a discrete outcome. Or you could do a cohort study where you assemble a large group of people, follow them for a long period of time, and at the beginning of the study, you do a lot of measurements. During the study, you do a lot of measurements, and you can look at a variety of multiple phenotypes, not just a single outcome. The traits, as I mentioned, can either be binary, that is discrete, or they can be continuous, so-called quantitative traits. And each provides different challenges and opportunities. And in most of these population-based studies over time, as many of you know, there's a blood draw. And with the blood draw, white cells are collected. Genomic DNA is potentially available. And in many of these studies, white cell pellets have been sitting around for, if not decades, years, waiting for genome science to get to the point that we'd be ready to use these resources to address interesting and important questions. And I was one of those people waiting, along with many of my colleagues. And fortunately for me, I've had an opportunity to use some of the white cell pellets that the lab generated for our studies. So in terms of cardiometabolic traits, what I wanted to point out here is this is an incomplete list, but just these are some examples. And I wanted to show you sort of quantitative traits like blood pressure and the discrete or binary trait hypertension, lipids, obviously total cholesterol, HDL, LDL, or LDL subclass or whatever, and then hypercholesterolemia, glucose, insulin, and diabetes, body mass index, waist, and obesity, uric acid and gout, coronary calcium and myocardial infarction, LV structure and function by echo or MR, and heart failure, QT interval, and sudden cardiac arrest. These are just examples of where you have quantitative traits that may relate to what we used to call diseases or risk factors, and now we increasingly call phenotypes that are complex. And there are many, many more. And in some of the work I'm going to show you today, you could add another 50 to the list of quantitative traits and another 30 to the list of complex traits. So genome-wide association studies basically are unbiased search, represent an unbiased search for novel loci. It's also described as an agnostic approach because it doesn't require any really thinking or planning up front in terms of knowledge of biology. There are about 10 million variants in the human genome, and they're inherited in blocks or haplotypes. And there's thought to be one single nucleotide polymorphism for every 300 base pairs. Now, of particular importance to what I'm going to show you is the fact that you can use one single nucleotide polymorphism to serve as a proxy for multiple or many SNPs. So you don't have to measure every SNP in the genome. You can basically get an approximation of what that SNP would be because it's highly correlated with another SNP, which we call a tag SNP. And this is a picture of a gene, and you can see that there are exons in the gene, there are introns in the gene, there's a 3' M, a 5' N, there's a promoter, and then there are places where you can have splice sites as well. You can have a SNP that's in an exon, you can have a SNP that's in an intron, or that's in a promoter. You can have a SNP that's not in a gene but is near a gene, and you can have a SNP that's in something called the gene desert, where there are no known genes near it. So keep in mind that when we're talking about SNPs in these studies, we're talking about tagged SNPs that are not picked because they're functional or because they're biologically important, but they're picked basically to represent different regions of the genome, okay? And one of the ways we're able to use this information, this tagged SNP approach, is because of linkage disequilibrium. Now, this basically shows you the LD, the linkage disequilibrium, in a gene with a large number of SNPs in that gene. And these are tagged SNPs. And basically what it shows you, if there's a very red box here, it means that SNP is in high linkage disequilibrium with another SNP. So you don't have to measure both SNPs, the variation in both SNPs. If you measure the variation in one, it gives you information about the variation in the other. If you see boxes here that are pink, it means the linkage disequilibrium is modest. And it may be that a particular tagged SNP doesn't really reproducibly estimate what the other SNP would be. And if they're white, there's no linkage at all. Now, what's happened is that because of this process of linkage disequilibrium, it turns out that not only do you not have to measure all of the SNPs, all 10 million, but you also can use the information you have to impute what a SNP might look like that you didn't measure, that you didn't genotype. And that's something that we've taken advantage of in genome-wide association studies. And I'll explain more in a minute. So there have been a number of challenges in genome-wide association studies. One is the sort of a bioinformatic challenge in that different platforms have been used to assess these tagged SNPs across the genome. Affymetrix has platforms, Illumina has the platforms, they have different versions of the platforms. You know, over time, they increase the number of SNPs on the platforms. And as you can imagine, different studies might use different platforms. And that's fine if you can get away with just looking at the data in your study. But we quickly learned that that was not going to be possible if we wanted to address the questions of whether genetic variation across the genome was related to these cardiometabolic traits. So you can have directly genotyped SNPs, and Affy has platforms of 1 million, which actually give you maybe 650,000 useful SNPs. They're working on platforms of 5 million. There are platforms, an Illumina platform of 370,000 SNPs, 500,000 SNPs. Illumina now has a 1 million SNP platform. So you can have those directly genotyped SNPs, but then you would like information on other SNPs. And what's happened is that because of the HapMap project, which was an effort to characterize the association of different SNPs with each other in these blocks that are inherited, it's been possible to impute 2.5 million SNPs from 370,000, 500,000, 600,000 SNPs that you actually genotype. So you basically have the genotyped SNPs, and then you have the imputed SNPs. And the imputation value, that is the confidence in the imputation, may be very, very high because the LD is very high, or it may be very modest because the LD is very low for the SNPs that are actually genotyped. Now, the reason this is important is because it became clear fairly early that we would need to pool findings from across multiple studies to be able to examine the associations of common variation of the genome with common cardiometabolic traits. And one of the challenges, besides the bioinformatical challenge and the statistical challenge, which I'll get to in a minute, was sort of an IRB challenge. How do you share these data? And some very clever people, including Thomas Lumley, Ken Rice, and others from our Department of Biostatistics here, had a very important role in defining the fact that we didn't need to share the individual data across these studies. All we needed to share was the findings within the study. And we could do a meta-analysis of findings as opposed to a meta-analysis of pooled data, pooled individual level data where you have the genotype and the phenotype for individuals. So the meta-analysis of findings was one way to finesse and get around the IRB constraints. But the other thing was the meta-analysis of findings was possible because of this ability to impute. So even if different platforms were used, you could all impute out to the 2.5 million SNPs in the HapMap, have a common metric, and then combine data. Now, the statistical issue that I list here is called multiple testing, and that's because basically the way these data are analyzed is by paired t-tests. So it's basically one t-test at a time, and you're doing 2.5 million tests. So you can imagine if you're doing 2.5 million tests that you're going to have a lot of positive results, right? You know that probably better than most people in this health science center. So 2.5 million tests. It turns out about 1 million of these SNPs are actually independent. That is, they're not in strong LD with one of the other SNPs. So we had to take into account multiple testing lest we be plagued, if you will, by getting very excited about p-values that looked extreme, which actually weren't extreme given the number of tests. I'm going to review with you the general approach to GWAS efforts. The first goal of GWAS and the primary goal is discovery. And basically, as I just suggested, in genome-wide association studies, we pool results or findings from multiple studies. These studies tend to be restricted by race ethnicity. Initially, the first wave of studies focused on European Americans because that's where the data was. There's now an effort to do similar work in African Americans, and eventually there'll be an effort to do it in Hispanic Americans, and probably last, Chinese Americans or American Indians. So the first set of efforts were restricted by race ethnicity. The modeling of these associations, that is looking at whether having 0, 1, or 2 copies, that is a additive model, additive genetic model, okay? 0, 1, or 2 copies of an allele, whether that having 0, 1, or 2 influences the level of a trait, let's say a quantitative trait. These analyses are adjusted for very few variables. They're adjusted for age, gender, and usually the study or the study site if a study has multiple sites. Occasionally, they're adjusted for covariates as well. But since there are very few covariates that can influence what your genome is, what your sequence variation is, it's not really a major challenge to take into account confounding, if you will, by other factors. The reason other factors are adjusted for in these analyses is usually to deal with 2 issues. One is population structure, which can be a source of bias, and the second is to try to improve the precision of estimates. And if you remove other determinants of that particular trait, you may improve the estimate of genetic effect. Population structure is taken into account by looking at whether there are principal components that reflect different subpopulations within your study population, and then you take that into account in the analysis. So you don't have a false positive association due to population substructure. Now, the second thing you do after discovery is you do replication. You really can't get a paper published in this field with discovery alone without replication or something else. Now, what I've done here is I've written as a goal replication, augmentation, and extension, because what's happened is that people have recognized that the only thing that can be replicated is a finding that's thought to be real from the beginning, that is from stage 1. And a finding that's thought to be real, you'll see, will need to have a p-value of 5 times 10 to the minus 8th. If you take 1 million tests and you divide it by 0.05, you basically get 5 times 10 to the minus 8th. That's the magic number, okay? So you have to have a p-value that's more extreme than that to be called genome-wide significant. If it's less than that, it may be interesting, it may be real, but it's not of genome-wide significance, and it needs to be demonstrated to be of genome-wide significance, possibly through augmentation, which basically means you add in a whole bunch of additional studies, either studies that have GWAS or studies that have DNA that can be used for genotyping, as I said here, of top hits. So that's how you do augmentation and replication. And then the second thing, or third thing you'd like to do is at some point you'd like to do transethnic comparisons, because you'd like to see if there's evidence in other race ethnicities that variation in that gene is related to the cardiometabolic trait of interest. Frequently, it will not be the same SNP, but you would like to see evidence that that gene is important in more than one race ethnicity. Now, having done the discovery and the replication, and by the way, what I want to say is if you do discovery and you don't achieve that magic threshold, and you have to augment it with the second stage, then you still have to do replication. Okay? Sometimes people will do a joint analysis of stage 1, stage 2. They find genome-wide significance. They think they're finished. They're not. They need to then show that that genome-wide significant hit with a joint analysis with a larger sample size, can be replicated in an independent sample. It's very important that the findings that are reported in literature are indeed findings that were not only discovered, but were replicated. So that it doesn't look like they're false positives, which was a real concern about the early work in genetic association studies, was that there were a lot of false positives. There were a lot of findings that couldn't be replicated. We've learned that lesson and we're dealing with it. So the goal of the third stage, if you will, is to go beyond GWAS and see if you can find some evidence that the association you identified with a tag SNP, which is not known to be functional or biologically important, basically can help you identify a causal variant in or near that gene or involved in that region of the genome. And in the old days, we used to say we were going to do fine mapping. We would do additional genotyping within the candidate gene or in the region. Now what we do is we go straight to resequencing of either you can resequence a targeted region, you can resequence exons, you can resequence regulatory regions, DNA hypersensitive sites, and so on and so forth, to try to see if you can determine what is the causal variant that might be linked, if you will, again, with linkage disequilibrium to the tag SNP association you observed. Now, I mentioned to you that we recognized fairly early on that we needed to pool the findings from studies, and that's because of something like this, these data here, which basically, these are p-values. This is 5 times 10 to the minus 8th, so it's the green line, okay? And this is where a minor allele frequency is 20%, and the relative risk estimate is 1.2 for a binary outcome. And here the minor allele frequency is 20%, and the relative risk is 1.3. Now, most of you wouldn't write home about a relative risk of 1.2 or 1.3. Those are pretty modest size effects. Smoking has a twofold increase in risk. A lot of diabetes may increase risk twofold for myocardial infarction and so on and so forth. Relative risks of 1.2, 1.3, we typically don't focus on because most of our studies aren't powered to find them. But that's what we thought optimistically we might find in some of these. And this just showed that the sample size that you needed, this is 4,000 cases and 4,000 controls. And this is the power. And you can see the power is modest even with 4,000 cases here. And that's in part because of the lower relative risk estimate than the relative risk estimate here. Now, what we then determined from these genome-wide studies, these consortia, was you really needed large sample sizes for discovery. And you needed data on both genotype and phenotype. You needed to be able to replicate in large independent samples. You needed either genotype data and DNA for the replication, and you needed collaboration and transparency. You needed principles by which people could work together across the globe and not be threatened and recognize that they're making a contribution to science and potentially to clinical medicine and the health of the public. And amazingly, this happened in a very short period of time. And it was in some ways a transformation. Recently, I received an email from a colleague at Yale who had not been involved in this work, but is a leader in cardiovascular outcomes research. And he was invited to participate in a GWAS, and he realized that within 24 hours, 24 studies with a population of 40,000 people had signed up to be involved in this GWAS. It took 24 to 36 hours. He sent an email off saying, "This is incredible. This is the way science should be done. I got to go tell my fellows what's happening." And the fact of the matter is, this has been happening for the last 2 or 3 years. And if you go to Nature Genetics, if you go to Nature, if you go to Lancet, if you go to Circulation Genetics, if you go to just a variety of journals, you will find examples of these kinds of collaborations. And you'll scratch your head and you'll say, why are there 300 authors on this paper? Why are there 30 authors on this paper? Why aren't there 5 authors on this paper? And the reason is that people realize that in order to contribute to science in this area, there's no choice. They have to work together. So one of the early examples of this was a study I was fortunate enough to be involved in with Steve Schwartz from epidemiology here and others, with colleagues from the Broad Institute and Mass General, with folks from Italy and from other studies. It was basically called the MIGen Consortium. and under the leadership of Say Cathreson and Chris O'Donnell, 6 studies were identified for discovery, 11 more for replication. The focus was on early onset MI. This was one of these examples where we had collected 600 cases of early onset MI, 300 in men under the age of 49 and 300 in women under the age of 59 from 3 counties in western Washington, King, Pierce, Snohomish, with a surveillance system of 38 acute care hospitals over 44 months. And we had this valuable resource, and we had white cell pellets. And the Broad, with Sake's leadership, did a discovery effort on approximately 3,000 cases and 3,000 controls of early onset MI. There was a replication in another 19,000 people over 4 stages. And this is one of these examples where the initial 3,000 didn't yield much. You augmented it with more, it yielded a little bit of something. You augmented it with more, it resulted in yet more. And then finally, what happened was you needed to replicate. This was an unusual example because all of the genotyping was done at the Broad. So it was, they used one platform, the AFI 6.0. This was published in Nature Genetics in February 2009. And at that time, we had identified 9 loci with genome-wide significance. The odds ratio for these loci varied from 1.1 to 1.3 for each copy of an allele. So there were 9 loci. Each loci has 0, 1, or 2 copies of the allele. If you have 1 copy of the minor allele versus none, your risk was increased anywhere from 10% to 30% for having an early onset MI. If you looked at MI genetic risk, combining these 9 loci, risk was increased 2.2-fold. John will tell us that if you ask about family history, it'll be associated with a 2.2-fold increased risk of MI. So 3 of the new loci in this study, 3 loci had not been previously described, 6 had previously been described by the Wellcome Trust folks. And interesting, there were no CNVs, which AFI thought we could measure, copy number variants related to this outcome at that time. Now we moved on from looking, I wanted to show you the MI gen data just because this talk is advertised as cardio-metabolic, and I wanted to at least have a little bit of cardio in it because the rest of the focus is going to be a little bit more metabolic. Again, a study like MI gen was an early study. We learned a lot, and it was an interesting study because we thought that early onset MI would be a genetically enriched population. Although we know that cigarette smoking is probably the most powerful determinant of early MI in men and in women. So what we did was we went on from that to say, oh, this collaboration seems to be working and it's important. So Bruce Pasady, who had an ancillary study to the cardiovascular health study, funded as an R01, Chris O'Donnell from Framingham, Eric Borwinkle from ERIC, and colleagues from the Rotterdam study and the Age Gene Environment study in Iceland, in Reykjavik, funded by the NIA, decided to pool their resources, that is, do meta-analyses of findings dealing with cardiometabolic traits in their populations to really get the sample size up to a level that seemed meaningful. And I want you to know that CHARGE, which stands for Cohorts for Heart and Aging Research in Genomic Epidemiology, and the University of Washington has been a major player in this effort, but it's not the only player, and there are over 300 investigators involved in CHARGE, and CHARGE is really now CHARGE Plus. There are probably many other folks involved in CHARGE other than those 300. These 5 studies are all cohort studies. Okay, the MI gen was basically case control because we were dealing with early onset MI. You can't really get enough cases if you do a cohort study for early onset MI. But for cohort studies, you can look at various metabolic traits, you can look at subclinical disease, you can look at risk factors, and you can look at complex traits as well. So the sample size here was 38,000. And if you take into account 3 other studies that we work with actively, Cardia, MESA, and Health ABC, the CHARGE Plus is now 53,000. And so each of these, the initial 5 cohorts used 4 different platforms, 2 AFI platforms and 2 Illumina platforms. And what was developed were standard measures for quality control and use of imputation to the HAP map and so on and so forth. And CHARGE looks at complex clinical phenotypes, at subclinical phenotypes, at molecular and physiologic phenotypes. And in some cases, these are related phenotypes. So, one would think, well, it's a chance to see whether if it's related to this phenotype, is it also related to that? And we try to avoid doing that because we know the phenotypes are correlated. And if you find an association with one, you're more likely to find the association with the other. So what we try to do is we want to know if a trait, let's say a gene's associated with uric acid, and we want to know if it's associated with MI. We try to look at an independent population for the association of that gene with MI, and not just look at it within the data, within the study that led to the association between the gene and uric acid. So I'm going to show you a few examples of the kinds of things we do. We do something called Q-plots, which basically what it does is it looks at the expected p-values here and the observed p-values there. And keep in mind, again, we're looking at 2.5 million tests. So we expect extreme p-values, but we don't expect more extreme p-values than you would get by chance alone. And that's what these represent. Okay, so if you see this is good, and this means that there might be something going on, because the observed p-values are more extreme. This is 10 to the minus 40th, or 10 to the minus 25th. It's more extreme than what you'd expect, given the multiple testing problem. Now, the other thing you'll see in these studies are Manhattan plots. Manhattan plots basically reflect the skyscrapers of Manhattan. And this is a hypothetical Manhattan plot. Just to orient you, these are the 22 autosomes. These are the p-values. This is the magic cut point. This is a series of SNPs associated with a single locus on chromosome 6. And these SNPs, many of them may be in LD, so it's not they're not independent tests. They may be pulled up here by something up here that's correlated with these other SNPs. These SNPs, while they look interesting, they would not be ready for replication. They'd be ready for augmentation if you want to do that, but they're not ready to be replicated because they could certainly be by chance alone. This is a hypothetical forest plot, which basically arrays the 5 charge cohorts. Each cohort has a sample size. It varies among the different cohorts. And you have point estimates in terms of this is the beta coefficient. And you have confidence intervals. If you looked within each cohort, none of them would be significant. If you look in a meta-analysis that pulls the findings from all of these cohorts, this beta coefficient, which is about minus 0.5, is significant in that the confidence interval doesn't overlap 0 there. This is a selected list of CHARGE results, and it reflects 15 of 68 publications prior to January 2011. And 24 of these publications were in Nature Genetics, and 2 of them were in Nature. And this is just to give you a sense of the range of phenotypes and the number of loci and the journals that these papers are being published in. Not listed here are phenotypes like calcium, magnesium, EGFR creatinine, EGFR cystatin C, many, many other phenotypes. I think we probably have adiponectin, you name it, anything that you could measure, and it could be coffee intake or fish intake or educational attainment. Any trait that you can think of, if it was measured in one of these cohort studies, and the cohort studies have thousands of measurements, we're in a position to determine, using this approach, whether there's any clue that variation across the genome might have some role in that particular trait. And we are running 24 hours a day in terms of analyses and paper writing. The other thing I would mention that's truly incredible is that most of this work is unfunded. Most of this work is unfunded. I'll say it again. Most of this work is unfunded, which basically means what has happened is that most of these papers, if you look at the papers, the first 5 people who are the lead authors and are starred are all young people. The last 5 people are all old people. People like me, who's old and just a cheerleader, I'm usually in the middle of, anywhere from 30 to 300 authors. And the fact is that of the 350 papers that was mentioned on my CV, probably 60 of them or 50 of them, I don't even know how many now. It's almost every month there's another paper that we're involved in. You can say, how can you be involved in all of these? Well, we contributed the data. We helped design the analysis. We reviewed the paper and gave critical feedback on the paper. We try to keep people from going beyond the data in terms of speculation, saying something is a loci when it hasn't been replicated or dealing with other issues. And so this is sort of an incredible effort. So one of the things you need to know is the loci that are identified in these studies. We call them top hits. They're not necessarily in genes, and they're not necessarily near genes, and they're not necessarily in regulatory regions. They can be, but they can be in all the above. When I showed you early on where tag SNPs are that are included in these arrays, they can be in exons, they can be in introns, they can be in promoter regions, they can be outside of the gene or near the gene, they can be in gene deserts. Maybe a third of the top hits are in exons, which means 2/3 are not. Whether they're in DHSs isn't clear. Whether they're in microRNA isn't clear. There are a lot of different possibilities by which these variations could be tagging a region that's important. So I have here known genes, previously unsuspected genes. Typically in these studies, what happens is we find the findings confirm the hard, painstaking, laborious work done by scientists, to identify pathways and proteins and so on and so forth. Invariably, we'll find something that's right on in terms of what's thought about the biology. But invariably, we'll find things that are previously unsuspected and may provide clues to new biology. And this could be, again, in coding regions or non-coding regions of exons. It could be in introns, or it can be in gene deserts. Now, one of the things that we have to deal with is that, and you'll hear this, and I mentioned it earlier, that basically all of this work, even though it's exciting and we found things, the reality is that the focus has been on main effects, on additive models, and on common variants. And basically what we have found is that common variants, by and large, have small effects. And the only way you can detect them is with these very large sample sizes. So you have studies now like the GIANT Consortium, which is the obesity consortium, that now has published papers with 250,000 subjects. 250,000 subjects to identify 18 loci. They've also published papers on height, where they identified 100 loci related to height. And so, keep in mind, we're looking at small effects. That means it explains a small proportion of the variation. The main effect explains a small proportion of the variation in the trait. We haven't, we're beginning now to look at rare variants with sequencing, and the expectation is that rare variants will have modest to large effects, although I don't know that that's going to be the case, because I think linkage was by and large disappointing. And rare variants may also have modest effects. It may be that what we need to do, rather than genetic testing, with all due respect, is get people to stop smoking or not adopt smoking. So then you have the gene by environment interaction effects. Everybody's interested in gene by environment. Really hard to study. You can imagine if you need tens of thousands of people to find main effects, to find gene environment effects, you really need large sample sizes. We put in a proposal recently to look at whether saturated fat intake in the diet interacts with genes and GWAS to influence lipid and lipoproteins. Scouring the earth, we could find 128,000 people who could be in our proposal. That is, they have genetic data or DNA, they have lipid and lipoprotein phenotypes, and they have dietary data that would enable us to say something about saturated fat intake. Whether that sample size is enough to do it, I don't know. The reviewers won't know either. We can estimate power, but until you actually do it, you don't know. So gene environment, a lot of stuff's been done now looking at physical activity, looking at diet, particularly as it relates to some of the genes I'm going to get at in a minute. And then you have epigenomics. Now I'm going to quickly run through some slides that deal with findings from the MAGIC consortium and diagram. MAGIC deals with type 2 diabetes quantitative traits among non-diabetics. So it's glucose, insulin, hemoglobin A1C, and so on. And diagram is basically the GWAS of type 2 diabetes. And we had Charge here because Charge basically folded its activities in this area with MAGIC and diagram. And you can see the sample sizes. FHS stands for Framingham. James Meggs, who works at Framingham, major player in MAGIC and diagram, shared some of these slides with me and invited me to present them to you. So this is diagram. You can see it's a case control study with 42,000 cases, 98,000 controls. And these are the major findings from MAGIC and diagram, and I'm going to walk you through them fairly slowly. This is fasting glucose here. This is the Manhattan plot. These are the top hits that basically exceed the genome-wide threshold. It's the blue line there. These are all of the loci you can see that are above that threshold for fasting glucose. Again, in MAGIC, fasting insulin, there's very few top hits that exceed genome-wide significance, in contrast to fasting glucose. If you look at 2-hour glucose, again, you see some of the same hits you see for fasting glucose, like GCKR, for example, is related to both of these traits, and TCF7L2, for example. So this is basically at the time they published this, which was in 2010. Jose Dupre and Richa Saxena published this. Both of these folks work in Boston, Jose at BU and Richa at MGH. When they published them, these were 14 genetic loci associated with fasting glucose, 2 with fasting insulin, 5 with 2-hour glucose. 9 of them were new, and 6 of these loci of the 14 were also associated with type 2 diabetes. Now, the GCKR is thought to be a functional SNP with a pleiotropic focus. It's related to many different phenotypes that we've looked at, including recently we published a paper on non-alcoholic fatty liver disease, and it was related to that as well. It's also related to fatty acids that we've looked at. The beta cell zinc transporter seems to be interesting in some of these studies and interacts potentially with dietary zinc. There are transcription factors. There's FADS, fatty acid desaturase, which is involved in fatty acid metabolism, and many other findings here, which I, in the interest of time, won't focus on. This is the Manhattan plot for type 2 diabetes. Again, many hits. This basically says this locus was established previously. That is, the red ones. The green ones were identified in this current study. And the blue ones were ones that had been identified in previous studies but were not confirmed in this study, which is the largest study to date. The interesting thing is, I don't know if I've ever presented a cartoon with pathways in it in a presentation in 30 years in academic medicine. So this is a first for me, and I got this slide from James. But the point is that in these analyses, one of the fascinating things is that a lot of the genes that are either, the SNP is either in the gene or near it, a lot of the genes that seem to be involved in these glucose homeostasis / diabetes traits are in beta cells. And all of these that are in blue here were related to one or more of the traits that was listed above. And it sort of raises some interesting questions. Hemoglobin A1C was another interesting one that we did. And basically, this was an analysis of 46,000 people of European ancestry. Again, you can see on the Manhattan plot, quite a few of the loci exceed genome-wide significance. And here we're talking about p-values of 10 to the minus 20. Very low probability that this difference was observed by chance alone. But these were also replicated. All of these were replicated. So 4 of them were known loci. 6 of them were novel, however. And interestingly, of the 6 novel loci, 2 affected iron metabolism, and 4 were associated with rare variants that are known to cause hereditary anemias. Now, this is hemoglobin A1C. This is the standard now for the diagnosis of diabetes. This is what reflects glucose homeostasis, but it also reflects iron metabolism, and it also reflects anemia. And while these effects may be small, they certainly were unknown before this GWAS was done. This is another cartoon, my second cartoon, just to give you an idea of the different tissues that might be involved. Now, this is a slide from James that suggests that genetic systems biology and type 2 diabetes may be informed by some of this work. So what you see down here, John, is predisposition to type 2 diabetes. And you see insulin resistance and reduced insulin secretion. And you work your way back to reduced beta cell mass, beta cell dysfunction, obesity, insulin resistance due to obesity. And you basically find different genes that may be operating through different pathways. So what do we do in follow-up? Well, first of all, we need to confirm these findings in other ancestry groups. Everything I've shown you today is in Europeans. Work is now ongoing to do trans-ethnic work, as I said, first in African Americans, later it will be done in Hispanics, then in Chinese Americans. We need to look for gene-environment interactions, but it's going to be really hard, and gene-gene interactions may be even harder, although there are efforts to take into account what we know in biology in terms of pathways. Obviously, functional genomics and seeing whether these SNPs or what we think are causal SNPs influence gene expression is also of importance. And then sequencing studies are now going on to follow up on the GWAS findings to see if we can identify either rare variants in those genes or causal variation. So in summary, multiple alleles with modest effect sizes have been associated with a wide range of cardiometabolic phenotypes or traits. And I think the surprise is not that they're related to metabolic traits. That's what metabolism is. It's incredible, but when you look at a metabolic phenotype like magnesium or uric acid or calcium, you find these p-values of 10 to the minus 58 or 10 to the minus 120 because metabolism is metabolism. And if variation in the genes that encode the proteins that influence that trait, whether it's a calcium sensing receptor or whether it's a uric acid transporter or whatever it is that you're close to in terms of your hit, is just fascinating. So it provides clues to novel proteins. uwvideo is a protein that was pointed to in our work on EGFR by a group that went from CHARGE. It's now called CKD-gen because it's much bigger than CHARGE. There's genetic evidence to support pleiotropy. Pleiotropy is what you're going to be hearing about at least for the next decade, if not beyond, which is that the same gene can influence multiple traits. Is that accounting for the clustering of the traits? Is that why when you lower a particular trait, you don't lower risk? Because it's not important in terms of the outcome of interest. It's just a pleiotropic relationship. That gene is also influencing other traits. Genetic risk scores explain a small proportion of the variance, and we shouldn't forget that. And then there's this issue of, well, maybe it'll be more important if we measure it early in life. And then there's the whole Mendelian randomization experiment stuff, which is if you can't show that genes that encode the proteins that influence the traits influence the outcome, then maybe the trait that you've been measuring, like CRP, is not causally related to MI. So I would close by saying that GWAS is only an initial step. Most of us who are doing it know that. Many people who don't do it don't think we know that. It's an initial step. Discoveries from GWAS, I think, do raise novel biological hypotheses. Interdisciplinary research collaborations are needed to explore further the mechanistic hypotheses raised by these studies and to translate them into clinical practice. So what have we learned? In February 2009, I gave a talk in Jerusalem on the transformation of genetic association studies reflected by the early work in GWAS. And I said something to the effect that population-based genome-wide association studies have successfully identified multiple loci with multiple cardiometabolic traits, and I said it was due to advances in science, technology, and social or global networking. And I predicted in that setting, it was a medical grand rounds, that there were going to be many papers over the next 3 or 4 years, and people should at least be aware that this was coming and what it might do and what it might not do. Now, I'm not a prophet, but in May 2011, If you ask me what we've learned that while these findings have the potential to lead to new biology in the short term, the long-term implications of the findings for clinical care remain to be determined. And I'll just mention, Harvey said I'm an attending. I'm actually a primary care provider. I have 100 patients at Harborview that I take care of. And so I'm on the front lines at least 6 hours a week to try to address their healthcare needs. And I think they really should be doing things other than worrying about whether they need to have their genome sequenced or have some panel of genotypes done at this point in time. So I want to thank, there are just way too many people to thank, but these are people, the first bullet are people who work with us at the Cardiovascular Health Research Unit, and the second bullet are some of the leaders in the field who we've worked with from other institutions. So with that, I'll close, and thank you very much.